So a warm welcome to everybody joining us for the third session on the first opening day of our this three-day event that we've put together with our partners and stakeholders from across the world. Thank you very much to the organizers and the behind the platform, the Adverse support team as well for this. Welcome to everybody joining us from across the world. This particular session is going to look at dengue, biomarkers, pathogenesis, and establish new horizons for potential dengue therapeutics and diagnostics. What's been really evident in the previous sessions that we've seen is the massive complexity when it comes to the immune landscape post-bite. So really we're talking about the kind of the way that immune landscape is informing treatment strategies, clinical management, as well as opening windows for potential new drug targets, earlier biomarkers for better diagnostics, and certainly the need for those has been emphasized by the previous speakers, Professor Tiki Pangestu's session in particular, as well as Professor Rosario's session to kick the things off in the morning. So I think the need is definitely there, and certainly COVID has really highlighted this whole COVID situation, the need for a more diverse approach to the way we look and get a feel for the mechanistic understanding behind infectious disease, and especially within dengue, this is true. The underlying mechanistic causes in terms of the dominant clinical features of severe dengue, and by that really I'm referring to the transient increase in terms of vascular permeability, really the vascular leakage, they remain to be better understood. And really the principle behind that, developing better understanding of that pathogenic, the landscape as it were, is that those deeper insights will give us improved treatment strategies, and it will obviously uncover, as we said, novel drug targets that are needed outside of the vaccine world, and also give earlier biomarkers that could be used for those diagnostics that are in urgent need. So I think leading the charge in many ways in terms of this particular research field are three of our panelists that we've the honor and pleasure of welcoming to this session. The first of those speakers is going to be Dr. Gauthari Nilika Malaviji from the University of Colombo in Sri Lanka, working closely with the Sri Lankan Ministry of Health, as well as collaborations with Oxford University as well. I think that's kind of more than enough. I think for me, I think we're kind of wary of the time. What I would ask is that there will be a Q&A at the end of the session. We're being joined by lots. We've had over 2,000 registrations in total in terms of the 3 days. I know we've got quite a few people here for this particular session. What I would say is please feel free to put your questions forward. I will try my utmost to channel those into the Q&A at the end. And if you could add your name, your country, and your affiliation to that question, it would make it easier in terms of a point of reference. That would be the only kind of housekeeping rule that I'm going to mention. I think that's enough from me, and I think it's time to hand over the floor to Dr. Gauthari Nilika Malaviji. So Nilika, over to you.
So welcome to this session on the pathogenesis and biomarkers. I'm from the University of Sri Jayawardhanapura, Sri Lanka, and I'll try to talk a bit about the pathogenesis and biomarkers of dengue. And I don't think it's, I should be talking about the global burden of dengue in this forum, of course, given that this is for the World Dengue Day. But we know that about 390 million infections of dengue occur annually. And the important things I would want to highlight here is the disability and the mortality. So dengue is seen in the tropical and subtropical areas as shown in this map. And the Southeast Asian region and the Latin American region are heavily affected by dengue with very high numbers of case loads. But when it comes to mortality, for some reason, the mortality rates are higher in the Southeast Asian and South Asian region compared to the Latin American region. despite high caseload in both areas. And that is something we need to look into, why we get this difference. And apart from mortality rates, the burden of dengue, the disability caused by dengue, is again more in the South Asian and Southeast Asian region compared to the Latin American region. Given that so many people are infected by dengue and it results in such a massive burden in especially resource-poor countries, dengue was named as one of the top threats to global health by the WHO in 2019. Now, I come from Sri Lanka, and we've had dengue epidemics annually, biannually since 1989, except for 2020 for some reason, with the emergence of COVID and maybe the restrictions, social distancing, and other measures taken. 2020 was a very quiet year for dengue. But as you can see, in Sri Lanka, if you take from 2006 to 2017, dengue has increased, the number of cases have increased along with wider geographical spread. And in Sri Lanka, the number of cases, the proportion of rising cases has been especially higher in old individuals compared to young individuals, the mortality and the morbidity. And of course, we don't know why this change in this age pattern is there, but the morbidity and mortality rates are highest in individuals between 20 to 40 years of age. And proportionately, the caseload is lower in school-going children compared to adults. And things have not been good over the years, and especially in 2017, we saw a really large dengue epidemic caused by the dengue 2 virus. And Sri Lanka is a developing country, so it is usual for our hospitals to always be full. And in dengue seasons, we have 2 patients per bed and 4 patients. But in 2017, because there was such a large number of patients, for the first time in Sri Lankan history, we had patients in corridors. So that's how bad it was in 2017, and much worse than the COVID situation. Now, as previous speakers in previous sessions have mentioned, the vast majority of dengue infections are asymptomatic, and which is most important. People don't show symptoms, they don't know whether they're infected. Some people do develop fever or symptoms, but those are not reported as dengue fever because those are not very severe. People don't come for testing or they go unnoticed. Another proportion of patients do develop dengue fever, and a minority develop complications of dengue in the form of dengue hemorrhagic fever and dengue shock syndrome. And then the WHO has categorized another clinical feature, which is expanded syndrome of dengue infection. So this is organ dysfunction in the absence of vascular leak or dengue hemorrhagic fever. Now, how do patients with dengue present? They present with fever, lethargy, muscle pains, joint pains, headache. Sore throat is sometimes seen in children, a large proportion of children, such as 25%. And patients with dengue do develop GIT symptoms such as abdominal pain, vomiting, and diarrhea. And if you do the initial lab test, would show lymphopenia, some degree of thrombocytopenia, elevated liver enzymes, and CRP may be elevated in dengue in some patients, especially those who go on to develop dengue hemorrhagic fever, but usually not more than 50. So when you look at these clinical features, and if you were not seeing the dengue here, I mean, they're very similar to the clinical features of COVID. So in countries where you have both dengue and COVID, you get there's a lot of issues with initial diagnosis, and I'll come to that later. Now, how does dengue manifest and what is the natural cause of dengue? So I just mentioned dengue starts with a rapid rise in temperature, and this high temperature phase is from day 1 to around day 3 to 4, and this coincides with the viremia. The patient has high fever and viremic, and as the viremia subsides, the temperature also subsides. But that is not good news for some patients. For those who develop dengue fever and who do not develop severe dengue in the form of dengue hemorrhagic fever, with the lowering of temperature, they directly go into the recovery phase, and that's it. But some patients who develop dengue hemorrhagic fever, with the lowering of the temperature, there is plasma leakage, and because of that, there is a rise in the hematocrit, and this coincides with the rapid drop in the platelet count. And this critical phase, where the fluid leakage happens lasts for 24 to 48 hours, after which the hematocrit rises, lowers because of fluid reabsorption into the capillaries, and it coincides with the rise of platelets, and the patients go on to recovery. Now, what is the pathogenesis behind this? And this is, of course, when the mosquito initially bites infected mosquito, it injects dengue viral particles into the intradermal capillaries, and these dengue viruses infect monocytes, macrophages, mast cells, dendritic cells, keratinocytes, many types of immune cells. And this initial viral replication results in the production of massive amounts of cytokines, chemokines, and other inflammatory mediators. And in this, after this rapid virus multiplication and production of these inflammatory cytokines and other mediators, these act on the endothelium, causing endothelial dysfunction. And because of this endothelial dysfunction, there is fluid leakage, which coincides with the critical phase. And when the patient, and when there is reabsorption of fluid and the endothelial dysfunction subsides, the patient goes on to the recovery phase. Now, plasma leakage, because of endothelial dysfunction, is the hallmark of severe dengue. And a lot of complications that occur during dengue hemorrhagic fever can be explained by the plasma leakage. So, for instance, because of the plasma leakage, there is accumulation of fluids in the pleural cavities and in the peritoneal cavities, causing pleural effusions and ascites. This leads to respiratory distress if they are very large. The plasma leakage results in hypotension because of reduction in blood pressure. Because reduction in blood pressure, there is poor organ perfusion, and this affects the liver, causing liver dysfunction, resulting in acute liver failure and hepatic encephalopathy. And poor organ perfusion of the kidney results in renal dysfunction. Because of this rapid lowering of the blood pressure, when it goes on without fluid replacement, that results in shock, and prolonged shock leads to cerebral hypoxia. And hypotension, when there is lowering of the blood pressure, the blood is shunted to important organs, and there is certain degree of intestinal and uterine mucosal ischemia, leading to gastrointestinal and vaginal bleeding. So plasma leakage is not the only reason of bleeding, but plasma leakage is an important cause of bleeding in dengue hemorrhagic fever. And some studies have shown that untreated dengue hemorrhagic fever is associated with a mortality of 20%. Now, the questions we have been interested in are what causes fluid leakage, and of course, why do some individuals only develop fluid leakage? Because the vast majority of individuals have asymptomatic infection, and the people who do show symptoms, the vast majority develop dengue fever, and it is only a minority, a smaller proportion, who develop severe dengue and go on to develop vascular leak and dengue hemorrhagic fever. And this just shows what is meant by endothelial dysfunction. So this graph, this is actually a picture of endothelial cells in the presence of normal healthy serum. And you can see the endothelial gap junctions are intact and they is nicely adhered to each other. But here are the endothelial cells in the presence of a serum from patient with dengue shock syndrome. And as you can see, there is gross degree of endothelial dysfunction. So this is what happens in dengue hemorrhagic fever and resulting in plasma leakage. So we have been trying to find out what causes this endothelial dysfunction and whether we can find ways to treat it. Now, there have been several mediators implicated in vascular leak. I have only listed some and some of the things that we've been looking at. So we have been especially interested in inflammatory lipid mediators, and I'll just show a little bit of data on that. PAF, the phospholipases, secretory phospholipase, and cytoplasmic phospholipase, and the leukotrienes. And cytokines, of course, have shown to cause vascular leak in other models, IL-6, IL-1 beta, TNF-alpha, and these cytokines come from different sources. There have been quite a bit of work done on mast cells, and mast cell products such as VEGF, tryptase, kinase, and histamine have shown to cause vascular leak. And NS1 alone, which is a secretory protein of the virus, which circulates independent of the virus, has also shown to cause vascular leak by Eva Harris's group. And of course, we've been looking at NS1 antibodies and if those are associated with vascular leak. Now, what we've been especially interested in is that one approach to treat dengue is antivirals, but whether another approach would be to find drug targets that prevent this vascular leak. And so during our investigations, we found that platelet activating factor, or PAF, is an important cause of vascular leak. So this is the pictures I showed earlier. And when you use a platelet activating factor receptor blocker, before adding the serum of the dengue, patients with dengue shock syndrome, there is a significant reduction in the endothelial dysfunction. So because of this data, we proceeded to do a preliminary study on the use of this drug called Predictive, which is antihistamine and also has a platelet activating factor receptor blocking effect. And so we did an initial randomized placebo-controlled trial. And in this trial, we found that people who were given Predictive had significantly less pleural effusions. The amount of leakage was less, and they were less likely to develop severe decrease of thrombocytopenia, and significantly inhibited or reduced the rise in the liver enzymes. And because of these initial preliminary results, we went on to do a subsequent study, a follow-up study, which was a double-blind, placebo-controlled randomized trial. And the main endpoint of that trial was the efficacy of Predictive to reduce the incidence of dengue hemorrhagic fever. And we had almost finished the trial, which just short of 19 patients when COVID happened. So we had to prematurely stop the trial, short of 19 patients. And just talking about the initial results of this trial, in the treatment arm, which consisted of 127 patients, the incidence of dengue hemorrhagic fever was 16.8, which is the placebo arm, which is the proportion of dengue hemorrhagic fever seen in patients receiving outpatient care in Sri Lanka. And in the Predictive arm, it was 9.8. So it did reduce the proportion of people developing fluid leakage, but of course we haven't completed the trial and we might just terminate it because there is no hope of continuing it. We have also been looking at other markers, especially because there have been publications, especially from Ashley St. John's group from Duke and US Singapore, about the mast cells in dengue. So we looked at this mediator called leukotrienes. So leukotrienes, the LT4 of leukotriene is a very stable compound in urine. So we looked at LT4 levels. And as you can see, the levels were significantly higher in patients with dengue hemorrhagic fever compared to dengue fever early in illness and later in illness. And when you look at the kinetics of LT4 in urine in acute dengue, they more or less remain the same levels throughout in patients with dengue fever, but significantly increased in patients with dengue hemorrhagic fever. So we're very excited that we could use this as a biomarker because it's a urinary substance, and it would be good to have a urinary test to look at a biomarker. But unfortunately, what we saw that there's a significant diurnal variation in the LT4 levels in patients with dengue fever and dengue hemorrhagic fever, and there's a several-fold difference in the morning versus afternoon, and by the way around in both dengue fever and dengue hemorrhagic fever. And we also saw, we did the viral loads of these individuals, and the viral loads also significantly changed in during morning and afternoon samples. So having looked at LT4, so one reason the reason we looked at LT4 was because there are already existing drugs to block leukotrienes, but another mediator we looked at is histamine, which is again a mast cell mediator, and of course there are antihistamines around which are widely used. Again, we saw that histamine level, urinary histamine levels were significantly higher in patients with dengue fever and dengue hemorrhagic fever compared to healthy individuals, although there were no difference between dengue fever and dengue hemorrhagic fever. So these studies have shown us that plasma leakage in dengue is the predominant cause of dengue hemorrhagic fever. And of course, apart from the mediators that I showed, leukotrienes and histamines, there are many other mediators that actually cause vascular leak, which have very conclusively shown to cause vascular leak. But PAF, LT4, and other mast cell products are important because there are existing drugs that actually block the activity of these mediators. So repurposing existing drugs should be explored, because there are very cheap drugs out there which known safety profiles that can be rapidly repurposed. Now, coming to the final challenges we have now, dengue was bad enough, but now we have COVID. And some countries have both, which are the tropical and subtropical resource poor countries. We know that COVID was seen in Europe, America, and certain countries last year. But of course, the vaccination rates are very high in those countries now, and Europe and America are coming out of COVID. But the vaccination rates in tropical and subtropical areas are very low, and they're likely to be low until early next year. So until such time, the dengue and COVID, the clinical presentation is similar. But the problem in dengue is patients we know develop rapid fluid leakage during day 3 to 7 of illness. And DHF mortality is very high, around 20% according to some studies, if untreated. Therefore, intense monitoring is needed. So in Sri Lanka, the mortality rates of dengue is like 0.3%. So it is, we have managed to reduce this to 0.3%, but that is with intense monitoring and timely fluid management. So, so it is very important to closely monitor patients with dengue, whereas such close monitoring or hourly or 4-hourly monitoring is not done in the case of COVID patients. So if dengue patients are misdiagnosed as COVID patients, or there's a diagnostic dilemma, that will have devastating consequences for patients. So, and as I just mentioned, countries like Sri Lanka, India, and many South Asian and Southeast Asian countries are the current epicenter for COVID-19. And we also have dengue. So we have not so good times ahead as far as both infections are concerned. I would like to end by thanking all our collaborators. So I'm from the Center for Dengue Research at the University of Sri Java, and I would like to thank everybody, all my colleagues and my team members, my students working there. Professor Graham Wong, our collaborator from University of Oxford, our collaborators from UK and US Singapore, Ashley St. Jones and Ewan Ui, and our collaborators from the National Institute of Infectious Diseases. This is the hospital where we do our dengue studies, Dr. Anandivikrama Irangana Arangota and Dr. Damayanti Dampiti. Thank you.
Thank you very, very much for that, Nilika. Fantastic presentation. We're already getting comments coming through the chat and the Q&A. Great presentation. Some kudos to you in terms of the clarity with which you've the clarity you brought to that this particular area of pathogenesis and the implications. In your last slide, just previous to your last slide, you were mentioning what happens if you get the diagnosis wrong because of the COVID co-infection, and really underlying the importance of your work and how it can then further inform not just the novel drug targets, but also the treatment strategies and the diagnostics themselves. So we'd like to say thank you very, very much for joining us all the way from Sri Lanka and highlighting those things. That kind of is a nice segue into our next presenter, Dr. Kevin Maringa from the Pirbright Institute here in the United Kingdom. If I could give the floor over to you, Kevin, to say hello. We're very happy that Kevin can join us. He's also part of our dengue advisory group, and we've had the pleasure of having a session with him before a few months ago in terms of some of the work that we do at the International Society for NTDs. But I'll pass the floor over for dengue diagnostics, challenges and opportunities by Dr. Kevin Maringa. Thank you. Thanks.
Thank you, Kamran, and thank you for the opportunity to speak. And can I just check, can you see my slides all right?
Yep, I think we can see them, yeah.
Yeah, so as Kamran introduced, my research group is based at the Pirbright Institute in the UK, and my research group really is very interested in understanding the molecular basis by which dengue virus is transmitted by the vector, and also how it causes vascular leakage in patients. So really how it causes that really critical part of the pathogenesis in patients. And in terms of studying those aspects, our focus has really been on NS1. So this protein that's secreted into the patient serum, as Nilika so very nicely introduced earlier. So I'm going to talk about diagnostics today, but our work, I think, could also lead to some very interesting new therapeutic targets. So I'm just going to move on. So Neelik already showed you this slide of clinical dengue and how it presents. I just want to point out a few things that will be important for my talk, which is that the viremia in dengue actually precedes this critical phase where you get severe disease. And as Neelik introduced, there are many factors that contribute to patients potentially progressing onto this critical phase, which are not completely understood yet. But it is interesting that the critical phase comes after the peak of viremia, and I'll come back to that later in relevance to our work. Also, when we think about diagnostics, a lot of diagnostics target the virus or viral products, which of course will detect this viremia, and some target antibodies, which actually come up quite late after infection. So I guess the way I like to think about it is, what's the purpose of diagnostics? And I think there are multiple purposes for diagnostics. The most obvious perhaps is for clinical management. So really confirming that clinical diagnosis that the doctor will have made. And that's really where the diagnostics are at the moment. But I think it goes without saying that the field is very interested in developing new diagnostics that can help predict which patients will develop severe disease, so that we can better manage dengue patients in hospital and really focus on the patients that are at high risk of developing severe disease. There is another aspect to diagnostics, which is that they are important for outbreak monitoring and reporting, so that we can measure both the scale of outbreaks and also which types of dengue serotypes and strains are circulating, and so that we can understand the changing epidemiology of dengue for planning for the future. And so this is just a slide really to highlight the kind of current types of dengue virus diagnostic tests that exist. I'm not going into individual tests, but just the principles behind the various tests that are available. So on the one hand, we have tests where we can isolate infectious virus from patient blood. So if you remember, virus will be present in the blood before the critical phase. Some of the issues with this is that it involves very high training and also facilities for high biocontainment to protect the operators from getting infected with dengue when they handle the samples. And so this is not, always possible in all settings. But also when we think about clinical usefulness, these sorts of virology methods are actually very slow. And so they're not particularly useful for managing patients in the clinic, but they are very useful for outbreak monitoring and seeing which serotypes are circulating. So in the clinic, we have mostly, my understanding is that people are using detection of viral antigens, particularly NS1, to correlate with that viremic phase, and also the detection of viral nucleic acid. And these tests tend to be a lot easier to use, and they're commonly used. Depending on how nucleic acids are being detected, you might need some specialist equipment and training. And of course, we need to consider costs. So, there are some particularly resource-poor settings where actually some of these tests are not routinely used, at least due to resources available. And then finally, another way that one can detect dengue is looking at the serology. So looking at the antibodies that are produced, particularly IgM, which is produced earlier and closer to the point of infection. But if you remember, the antibodies actually come up still relatively late, and actually these sorts of serology are maybe a bit more useful in combination with other tests. And they're very useful for monitoring what serotypes are circulating in a region. So I've sort of listed here what features might be desirable in a predictive dengue diagnostic test. So all of those tests I mentioned to you before, I should have said, are really aimed at identifying which patients have dengue, but they don't actually help us predict which dengue patients will develop dengue hemorrhagic fever, severe dengue, and vascular leakage. So really, we would want to have biomarkers that help us predict progression to severe disease, ideally at the point at which patients already present in hospital, so several days before they will go on to develop severe disease. We would like those tests to have a high sensitivity and specificity so that we can really be accurate in our triaging of patients into high risk and low risk categories. And I think we have to think, of course, very carefully about the sorts of settings that we might be using these tests in, because they won't only be useful in hospitals where we have maybe more resources, but also maybe in more community and healthcare centers or resource poor settings. So ideally, these tests should be low cost, they should be easy to use so that you don't need particularly high levels of specialist equipment or training, and we should have supply chains that are manageable, for example, without cold chain management and that kind of thing. So Nilika also really gave a really nice introduction to the endothelium and how the endothelium is disrupted during a severe dengue. And so I'm going to talk a little bit about our work that we've been doing in this area to understand really how the microvasculature, how the capillaries actually open up to cause that vascular leakage. And so much of the field really has focused on these endothelial cells, which form the physical barrier of the capillaries. But this misses some of the complexity of what the microvasculature actually looks like, because you actually have another cell type also present in capillaries called pericytes or perivascular cells. And these perivascular cells actually, they don't form the barrier itself, but they actually interact with endothelial cells both through direct contact and through soluble mediators, soluble signaling molecules, and they're very critical for maintaining the barrier. So there are some non-communicative diseases where pericyte function is disrupted, and that will also cause vascular leakage, even if there is no direct effect on endothelial cells. So they are really very critical for maintaining the microvascular barrier. And so we set up a really great collaboration with Dr. Paola Campagnolo at the University of Surrey, and most of the work, or all of the work I'll present today, was done by my PhD student, Yin Chung. And so I'm just going to present some of our work of looking at these parasites and how they're affected during dengue. And I think the first thing to say is, so Nilika showed some really beautiful slides of how you can actually look and see those endothelial cell barriers opening up when you add dengue serum, for example, or otherwise treat them. So we've used a slightly different assay. So here what we're doing is we're growing these endothelial cell monolayers on a membrane, and we can measure what we call the transandrothelial electrical resistance. So we can measure the difference in the electrical potential across this membrane. And if the monolayer is very nice and tight, like you would expect for the microvascular barrier, we're going to have a nice high electrical resistance, which we see here in the bold line. If we add NS1 or vasodilatory cytokines like TNF, this will actually cause the barrier to start opening. And what you see is you see a dip in the electrical resistance. So you see as this resistance goes down, this is indicative that these monolayers are actually starting to become more leaky and that molecules are able to go through them. So this is a direct measure of vascular leakage, albeit in this in vitro cell culture system. And so, as Nilika introduced very nicely, many other groups, or several other groups, including Eva Harris's group, have shown that the viral protein NS1 alone will actually cause some form of vascular leakage. This is obviously in a patient in combination with the cytokines that are produced and all the other molecules and NS1 antibodies that also have effects. But we've really started by studying just the effects of NS1. And here I'm showing you a different kind of assay. So we actually decided to look at, so that you can grow these endothelial cells in monolayers, but what we're doing here is we're growing them in actually a more complex substrate that allows the endothelial cells to form capillary-like structures in 3 dimensions through this substrate. And so this actually looks a little bit more like what a capillary would look like in vivo, as opposed to these monolayers in a dish. And when we have here endothelial cells alone, we see the formation of some capillaries. And when we add pericytes, so these really crucial support cells, you can see that there are a greater number of branches, a greater number of these capillary-like structures, and these capillary structures are getting thicker. And that's because the pericytes are supporting those endothelial cells and their critical function, and they're helping these capillaries to form. And then we can treat these cultures with NS1. And what you can hopefully see is that actually you start seeing that the branch width, for example, is getting much lower, and these endothelial capillary structures are becoming disrupted as NS1 is disrupting this interaction with pericytes. So this was a very interesting finding, showing for the first time that NS1 disrupts this sort of actual interaction between these 2 cell types which is so crucial for the microvascular barrier. And we can quantify that in various different ways. And here we're looking at the width of the capillaries. And what you can see again here is if you just have endothelial cells, we have a certain width, which we normalize everything to. If you add parasites, you increase that width. So you increase this formation of the capillaries and the barrier. And then you can treat with increasing amounts of NS1. And you can see that the more NS1 you add, the more likely you are to disrupt this interaction. And I think a really important point to make here is that a lot of these studies with NS1 have been done at very high concentrations of NS1 in these cell cultures in other research groups. You do see concentrations of NS1 in this range, but when you look at the spread of NS1 concentrations, even in patients who develop hemorrhage, you can actually have patients with relatively low concentrations of NS1 still develop hemorrhage. And so what's really unique about our system here is that we also see leakage at these lower concentrations of NS1, which are a little bit more reflective of the full range of NS1 concentrations you would see in a patient. And this is going to be the last data slide I show you, but we can then do some other interesting assays. So here we've gone back to our transandrothelial electrical resistance assay, where we culture our endothelial cells on the monolayer in this transwell insert. And then we actually have co-cultured these with parasites. And what's important in this assay to understand is, if you remember, parasites can communicate with endothelial cells through direct contacts, as they did in our three-dimensional culture, but also through signaling molecules that are soluble. And those signaling molecules are much more interesting when you think about developing biomarkers, because you might be able to detect those in the bloodstream. And so here we've set up an assay where you can actually, the cells are not in direct contact, but they can signal to each other. And again, you see that the parasites actually increase the strength of this barrier. So here we have endothelial cells alone. You add the parasites and the barrier is strengthened. And if you add NS1 again, or TNF as our vasodilatory cytokine control, you see that NS1 is completely disrupting this ability of parasites to help endothelial cells form this barrier. And I think what this is indicating is that NS1 is actually disrupting soluble communication through soluble mediators between endothelial cells and parasites, potentially very early in infection. And so we're very interested in developing this further to see whether we can use this to develop biomarkers of severe dengue. So I'm just going to finish with this slide. I'm sorry. So we think we've come up with a slightly, we think we've come up with a slightly more improved model maybe of NS1 mediated vascular leakage. So in a healthy person, we have our endothelial cells forming the microvascular barrier surrounded by parasites, which are helping support critically that barrier formation. During that, during that dengue virus replication, you're getting the production of these inflammatory cytokines and all these other molecules, including NS1, that will have direct effects on the endothelial cells, as Neelika introduced and many groups have shown. And we think what happens then is that you get some opening of the vasculature, but we think that actually really the big opening, that really like critical vascular leak, actually happens a bit later. And if you remember, the critical phase actually comes after this peak of NS1. So that initially opening of the vasculature would allow NS1 and potentially other cytokines and other factors to then escape from this capillary, start having effects on parasites, and then start affecting the way that parasites are able to support endothelial cells. And we think this actually causes a delayed, but then the very big vascular leakage that you see after the peak in dengue replication, through some effect on soluble mediators between parasites and endothelial cells. And we think that actually trying to understand what molecules are disrupted by NS1 in parasites is very interesting for trying to understand and maybe identify biomarkers, but also maybe some new therapeutics that help to prevent this leakage. And so thank you for attention, and I'd be very happy to take questions after the next speaker.
Thank you very, very much for that, Kevin. Some of the very complex interactions in terms of your own work and how that is going to go ahead and ultimately inform a better world for diagnostics, the importance of it. and some of the kind of interplays there between the various phases along in terms of the evolution of dengue post-bite. So thank you very, very much for that. We're having some questions come through from the Q&A. I know you're coughing there. People can't see this because of the virtual backgrounds, but it's a very hot day here in London. So I appreciate the fact that you battled on through that. It's very rare that we have such good weather, and it's an excellent day today. So I do sympathize with your dry throat, but thank you very much for battling on. So 2 a.m. over in California. We're brings us to the next speaker, Dr. Priya Shah from the University of California, Davis. Priya, thank you very, very much for joining us so early in your day. And I guess the floor is yours. Handing over to yourself.
Thank you very much, Kamran, for your invitation and for the introduction. I'm really excited to be here, even though it is pretty early in the morning. So let me just find my slides here.
Yeah, we can see them. We can see them.
All right, great. So my group, very similar to Kevin's group, is focused on these molecular interactions for dengue virus. And what we're specifically interested in is these molecular interactions between the virus and host proteins that might reveal interesting aspects of virus replication and disease. And so what I'm going to tell you about today is some of our work, but how that might relate to identifying novel antiviral therapeutic strategies. And I'll give you one example of some of the work that we've done in this area. Okay, and so I think Nilika really nicely outlined the dynamics of dengue disease and how viremia happens early along with the fever, and then you get the severe disease following on that. And so both Nilika and Kevin's group have done some really exciting work related to treating that disease, but also diagnosing that severe disease. And what I want to emphasize is that what I'm going to be talking about in this short talk is dealing with some of the molecular level events that happen during that viremia phase, that virus replication phase, that could be targeted therapeutically. Okay. And so when we talk about these types of antiviral compounds that actually target the virus and inhibit virus replication, typically these are small molecule compounds that target a viral protein. And so I'm giving you one of the exciting new examples of these antiviral compounds for dengue virus in particular that was not published by my group. So this is work done by some very talented scientists that was published in Science Translational Medicine earlier this year, some very eminent dengue virologists and clinicians. And what they identified was this chemical compound called NITD 688 that targets a dengue virus protein called NS4A, or non-structural protein 4A. And that is shown right here. And it's shown right here in this cartoon. And so this chemical compound, it serves to inhibit the function of this viral protein, and that's how virus replication and viremia are lowered. Now, the issue with a lot of antiviral therapies that target viral proteins is this problem of drug resistance. And we've seen this through the early 90s with HIV until the combination therapies were developed. We have been worried about this with our influenza therapies that are very useful right now. And we've also been worried about this with SARS-CoV-2 and the emergence of not only variants that are more pathogenic, but also variants that evade some of our great tools in the clinic, like remdesivir. And so what you can see in this diagram here are 3 mutations that are indicated by these stars here that allow this dengue virus to evade or escape inhibition by the small molecule compound. And so these viruses, they're incredibly effective at evolving to evade any sort of selective pressure. And so viruses, especially RNA viruses, are going to rapidly evolve capabilities like this. Now, these viruses do exhibit lower replication capacities. They're not as capable of replicating as the wild type or original isolate in many cases, but viruses can always then continue to adapt and improve upon those escape mutations, right? And so this is one of the concerns that we always have in developing antiviral therapies. And so what I want to talk about today is a different approach to developing antiviral therapies, where we target not the virus, but the host. Okay, and the reason we're going to do this is because all viruses have to interact with and hijack host cellular proteins to replicate. So now what I want you to do, we've been talking about, this sort of organismal and tissue level disease. What I want to do is zoom down to the molecular level and talk about what's actually going on inside of a single cell for dengue virus to replicate. And if you think about it, any virus, including dengue virus, it has to do three things. It has to get inside the cell, it has to replicate its genome, and then it has to get out of the cell, okay? And to do all of these tasks, viruses interact with host proteins through physical interactions, which we depict through these cartoon diagrams where the square in the center is a viral protein, and all of these circles are human proteins that viral protein physically interacts with, is connected to in a very physical sense to facilitate these very basic tasks of virus replication that have to happen inside a host cell. And so with dengue virus, because it's a mosquito-borne virus, these basic aspects of virus replication, they have to occur in both the human host and the human cells and mosquito cells. And so one way of targeting the virus where it really hurts is to identify these virus host protein interactions that are essential for replication in human and mosquito cells and go after those essential features to inhibit the virus. And so that's exactly what we've done with dengue virus host protein interactions. And so what we did is we identified these virus host protein interactions that I'm showing as a cartoon here for every single dengue virus protein in both human and mosquito cells. And we did this by purifying the viral protein in either human or mosquito cells, and then analyzing all of the host proteins using mass spectrometry. Okay, and so I'm glossing over many years of work when I show you the cartoons here that summarize the data. And what you're looking at here in the blue are all of the dengue virus human protein interactions that we identified, that we found to be the most significant. And there are about 200 of those. And what you see in the gold on the right side of the slide is all of the dengue virus mosquito protein interactions that we identified to be high confidence. And there's about 150 of them that we identified. And so what we can do is we can use a lot of bioinformatic network integration approaches to basically combine these two networks and identify what's shared between the two. And we can do this at 3 different levels. We can do this with individual proteins. We can do this with entire protein complexes. And then we can do this with biological pathways and processes in the cell to see what's the same and what's different. And so that's what I'm showing you here in this slide. And so if you focus on the diagram on the left side of the slide, what you're seeing is that integration of all the protein interaction network data for the human proteins, which are in blue, and the mosquito proteins, which are in this orange color. And so here I'm showing you data for just one viral protein called non-structural protein 4A. And I'm showing you this data in particular because it was really exciting to us. What we found were that, first of all, this protein had a lot of shared characteristics in the human and mosquito cells in terms of the types of proteins it interacted with. It interacted with host proteins that were involved in oxidative phosphorylation. So that's, our aerobic respiration cycle that eukaryotes take advantage of. We saw a lot of proteins involved in organelle targeting and organization, in glycosylation, because as we know with proteins like NS1, they need to be glycosylated to get out of the cell. And we also saw a lot of proteins involved in trafficking and potentially involved in that entry and exit of the virus from the cells. And so that's at the pathway level. We could also zoom in on individual proteins. And that's what I'm showing you in this panel here, where you can see the individual proteins that were either identified in the human cells or the mosquito cells that make up this category involved in organelle targeting and organization. And the proteins that I want to highlight to you are part of this complex called the Sec61 translocon. This is an essential complex found in all eukaryotes that is involved in transmembrane protein biosynthesis. And so dengue virus, like all other flaviviruses, have a lot of transmembrane proteins. And so taking advantage of the Sec61 translocon will help the virus translate and produce functional transmembrane proteins. And so what we decided to do was collaborate with a professor at UCSF, Jack Taunton, who specializes in studying this translocon, the Sec61 translocon, and the fundamental biology behind it. But what they've also done in the Taunton group is they've developed small molecule inhibitors of this complex. And so we took these small molecule inhibitors and used them to inhibit dengue virus replication in human and mosquito cells. And so here you can see what the normal virus replication looks like in both of these cell types. And when we add either inhibitor for the Sec61 translocon, that virus replication is essentially undetectable using our plaque assay methods. Importantly, one thing that you might be worried about, especially when you target a human complex, is toxicity to the host. What's exciting about these small molecule inhibitors is that they really modulate the activity of the translocon. They don't inhibit it outright. And so this complex still has the ability to synthesize many transmembrane proteins, including the ones that are essential for cell viability. It's just a select number of transmembrane proteins that are affected by these inhibitors, including flavivirus proteins. And so what you can see here is a measurement of cell viability for our DMSO dimethyl sulfoxide carrier control, along with the two inhibitors. And the cell viability is essentially unaffected by these inhibitors. What else is exciting is that we can do time of addition assays to show that even when we infect before we add the drug, if we add the drug at later points, we can still inhibit virus replication, which is what you see here with protein synthesis of NS2B, another viral protein that we can use to track virus replication at very early stages. And so this is one example of how these conserved interactions with host proteins have a really strong therapeutic potential for dengue virus, but also other emerging diseases or neglected tropical diseases as well. And what this can do is help treat patients in that viremia phase to help get them back into the workforce if they're simply suffering from dengue fever, because it's still a very debilitating disease, or even help prevent them from producing a more severe disease like dengue hemorrhagic fever and shock syndrome. Okay, and so with that, I'd like to thank the members of my group at UC Davis and also my collaborators, past and present, especially Nevin Krogan, my postdoc advisor, who I did a lot of this work with, and Jack Taunton, who created those Sec61 translocon inhibitors, and my funding sources. And of course, I'm happy to take any questions, and I think there's going to be a great panel discussion at the end of this.
So thank you very, very much for that, Priya. Fantastic and very complex subject area. I love the mapping of human interaction, the protein and the mosquito interactions to yield potentially new targets. Fascinating approach, and hats off to you and your team, your lab, for running that, and I'm sure it's kicked off with a few questions that have come through from the kind of audience as well, and I know that you've been kind of answering those in some form or another. My questions are really going to be centered around looking at this from the three kind of phases in terms of the evolution of dengue. So the early febrile phase, in which we've all mentioned there, the viremia peaks in that particular phase, host inflammatory response is rising, capillary leaking is starting but not peaking. You've got probably a high concentration of circulating NS1 going down, thrombocytopenia starting throughout this early febrile phase. So my question to you all, in this early phase, what would be the most promising, from your opinion, drug targets or diagnostic targets? I'll start with Nilika, if that's okay. So what would be the most promising targets coming through, or areas, avenues? I know you probably summarized it in some of the slides, but just for the benefit of the Q&A, what would be the most promising areas in the early febrile stage?
Yes, so actually, although the vascular leak is seen to occur between day 3 to 7, we see all these mediators that cause vascular leak, starting from host mediators like cytokines, inflammatory lipid mediators, the NS1 that Kevin showed. All of those, the highest levels are seen in the febrile phase. The NS1 is, of course, is a viral protein, but even the host cytokines or lipid mediators that result in vascular leak, like the ones I showed, PAF, and the enzymes that generate PAF, like cytoplasmic phospholipases, secretory phospholipases, IL-6, IL-1 beta, TNF-alpha, all of those are very much higher in the febrile phase than the critical phase, because we've done like twice daily sampling from the beginning of the illness as you proceed. So because of that, although it might make sense, okay, it would to think that only antiviral works during early illness, actually, because the mediators are, all these mediators are very much higher early in illness. I think the drugs that target vascular leak in the form of host proteins are ideally also given in early illness. So if I can elaborate on that, in the trials we did with rupatadine, and when we did a sub-analysis, when you gave rupatadine after day 4, which is when most people have sort of got vascular leak, it didn't do much. I mean, it did reduce, but it didn't prevent it. Whereas if you gave it very early, like within the first 3 days, it then prevented the proportion of people developing dengue hemorrhagic fever. So I think whatever intervention that it has to be done early. So in order to do an intervention early, public education is important to get down the patients early. Thank you.
Okay, thanks for that answer. Thank you very much. Are you aware, may I ask, are you aware of any kind of repurposing kind of programs, or kind of, looks into that, forays into that, using some of these PAF targets, these PLA2, the CPLA2 that you mentioned, these early markers in the early phase? Are you aware of any repurposing going on in that at the moment?
Yes, so we've been doing the rupatadine, and we've also been we had got ethics, and we were going to start Montelukast, which is widely used in, again, respiratory allergies and chronic urticaria. And we were just about to start that trial when COVID happened. But I know in Singapore, people are using other types of things like ketotifen, which again inhibits, is a mast cell blocker mediated master stabilizer. So I know Ashley St. Jones was doing a trial there. Then people are also using things like metformin, which is anti-diabetic drug. Trials are, I think, have started in Singapore. I know they were going to start. I don't know with the COVID whether that was on hold or not. So they were going to start metformin which is an anti-diabetic drug. Again, I think when we look at dengue, the proportion of individuals, especially in Singapore, Sri Lanka, and certain countries, it is more, the morbidity and mortality are higher, becoming higher in adults, and especially in people with diabetes. So, an endothelial dysfunction, taking lessons from COVID, endothelial dysfunction appears to be a problem in COVID. It is much more a problem in dengue. So whether people, the risk factors of developing severe dengue, I mean diabetes, and these metabolic factors have been shown in so many studies. So there are studies looking into repurposing anti-diabetic drugs as well. Thank you.
Fantastic. That's a great answer. Thank you very, very much for that, Nilika. I'll move to Kevin with the same answer in terms of this early phase. What would be your, I'm not really asking recommendations, but what would be the pointers? What's working, what's not working? What would you say is the most promising avenue in that early febrile phase?
Yes, I'm not a clinician, so I'm not going to probably step on Nilika's toes. She knows a lot more of what's being used in the clinic. I guess what's interesting to me in this parasite, in our study of parasites, is that other people, when they have looked in the clinic, some of these biomarkers that Nilika was talking about, we also see very early dysregulation of some markers that are known to be associated with this interaction between parasites and endothelial cells. For example, angiopoietin signaling is dysregulated in dengue, but actually in other viral diseases as well, where you see vascular leakage, for example, where viruses cross the blood-brain barrier or the placenta as well. So there is evidence that some of this signaling might be disrupted early and it might be useful, but we're currently trying to see whether there's actually other markers that we might not know about that might be specific to dengue. But I think when we think about the treatment, I suppose if we're looking, in my lab, we're very interested, obviously, in this leakage. And I guess one important point to think about when we think of treatments, if you sort of look holistically at the three talks you've seen today, is if we develop it, so if we were to develop drugs that block leakage, that actually doesn't stop people, getting very sick with dengue, albeit that they may not enter the critical phase. So I think, some of Priya Shah's work is very interesting because, of course, stopping symptoms, we often think about the critical phase in dengue and the leakage, but it's a pretty severe and horrible disease, even if you don't go into leakage. And ideally, actually, we would like to develop therapies against those, well, to stop symptoms to begin with.
That's a great answer, Kevin. Thank you very much for that. Do you anticipate because homing on the diagnostics of it, do you anticipate a new diagnostic coming out from your lab or from the work that you're involved in that would kind of bring all of this together? Kevin?
I mean, we're a very long way away from in my lab. I'm aware of other groups that are doing a lot of work in this area. I think where the field is at the moment is that it's difficult to find one single marker at the moment. There are various different markers that have been associated, but they're not necessarily associated enough to use in the clinic. And I know there's some interesting groups. Andrew Davidson, who's done at the University of Bristol, has done a lot of work in the Philippines and also with collaborators in Indonesia, actually looking at combining biomarkers, I think, and that might actually be quite fruitful. Because individual markers may not be as promising as maybe people had initially hoped.
Yeah, and I'm assuming, obviously I asked that question for a reason, that there's such a diverse range of attendees, not just now, but across the 3 days. You're open to collaboration at the Pirbright Institute. If people, that's kind of a rhetorical, well, that's a question to you, Kevin. If people want to get in touch with you about driving some of this forward with you, are you open to that? If, what's your outlook on that?
Yeah, so absolutely. I mean, I'm happy for people to contact me. And I would say, of course, we're doing this in collaboration with the University of Surrey, because the Pirbright Institute's really interested in zoonotic diseases and veterinary diseases and arboviruses, more in the context of the vector, which is actually mostly my work. So we're actually starting to move more towards some of these zoonotic hemorrhagic flaviviruses in my group, but we will continue our dengue work in collaboration with the University of Surrey in terms of the human disease.
Brilliant. Thank you very much for that, Kevin. Priya, I know that we left you to last because you were the last presenter there. I didn't want to overload you there at 2 a.m. You mentioned NS4A, NS4A and SEC61 in terms of this early phase, as far as I understood that, if I understood that properly. Are these that's your recommendation? Are there other areas around that? Or
no, I'm kind of the all of the above mentality when it comes to trying to find solutions, right? And so, you know, I would love to explore additional host targets, right? We went after SEC61 because people have known for a long time using functional genomic screens back from RNAi in the aughts to CRISPR in the teens, that SEC61 is important for flavivirus replication and it's essential, right? So we had that piece of information. We didn't know what it was doing for the virus, right? And so this physical connection was really valuable in understanding that it was potentially an early feature, an early event in virus replication. And when you're thinking about antivirals targeting earlier events in the whole replication cycle, those 3 steps that have to happen, that's always going to be better in terms of dealing with the emergence of viral resistance and also just producing better outcomes for patients, right? And so like if you can find an entry inhibitor, right, that would be great. And we've seen that with HIV, right? And so I think having that sort of very early step in virus replication, the first thing that has to happen with a virus like dengue virus, a positive strand RNA virus, is that it has to get translated to produce that protein. And so if we hit it where it really hurts right there, it's not going to be able to do any of the subsequent steps. And so that's why that one is really attractive. The fact that it's not toxic, even though it's hitting an essential host pathway, is promising. And so the Taunton lab is doing work to look at clinical applications for other diseases as well. And so I think it is a promising avenue, but I'm also really interested in understanding, you know, can we find modulators, not inhibitors, but something that can fine tune some of these pathways more specifically, like glycosylation, like mitochondrial activity, right? Because that could be really exciting. I think the other thing to remember is that, you know, by the time someone shows up to the hospital with dengue fever, right? Like, is there really a clinical window for treatment for that patient? I'm not a clinician, so I don't know, right? But I guess one of the things that I also really like to think about in terms of timing and applications of these types of antivirals that focus on the viremia side of things, as opposed to the vascular leakage side of things, is that if you look at some of the very granular data on dengue transmission amongst household members, right? Like the transmission is happening within the household or with nearest neighbors because the mosquitoes don't go that far. And so one of the opportunities might be if you have an ambulatory dengue fever patient that gets sent back home, maybe the caregivers and the neighbors should be given one of these therapeutics prophylactically. Right. And so I think there's a huge opportunity there as well to benefit those communities in particular that are hard hit year after year by this disease.
Great answer. Thank you very much for that, Priya. Really showing how your work can impact these treatment strategies that need to be reevaluated. So that's a great answer. Thank you very much for that. Just coming to try to bring some of the questions that have been asked into this a little bit. I know you've answered this, Nilika, personally to the person. I think Dr. Khurram Wahid was asking a question around repurposing drugs like antiviral drugs if around IL-6. Would this be beneficial or not? I know you had that up on your slide earlier as well, Nilika. Would you care to kind of give us a kind of bit of your insights into that repurposing drugs on IL-6 would be beneficial or not?
So IL-6 levels are high in dengue, but we did a comparison study about the cytokine storms in COVID versus dengue. And actually in COVID, the IL-6 levels are like about 20 to 100 times higher than you would see in dengue. So we know that the recovery trial has shown that, for instance, IL-6 tocilizumab does work in patients with severe COVID, but it has to be, the timing has to be right. And in dengue, IL-6 levels are high, but not at all to that level. And as Priya and Kevin just mentioned, it is not just one, but it's a lot of things coming together. And IL-6 is just one of them, because we know that just because you've got IL-10, you still have the other cytokines like TNF alpha and IL-1 beta. And apart from that, you have all the mast cell mediators and lipid mediators like PAF, leukotrienes, histamines, tryptases, chimases, which all of them do as well cause vascular leak. And apart from the host factors, you have the NS1 that leads to vascular leak. So just by blocking IL-6, I don't think, given, I mean, if it was the cytokine and you have very high levels as you see in COVID, then definitely, yes, I would say it's worth doing a trial. But when we did a comparison of COVID versus dengue, and IL-6 levels were nothing compared to in dengue compared to COVID. Thank you.
Great answer. Thank you very much. I know that you'd answered a question earlier as well. There was a question from Dr. Ida Safitiri-Laksono, complimenting the great presentations and also talking about the early phase. That's why I'm asking this question. She's talking about, where she mentions that in the real settings, many dengue patients presenting in the clinic may already be in the critical period as they present, i.e. day 4 to 5. And so did any of the drugs that you've seen or any of the kind of insights you got into those the stage, I suppose, the inhibiting viral replication as a potential area, release of mediators within that. Do you think any of those will have potential benefits, given that the patient will be presenting, they're probably already entering the critical day 4 or 5 critical period post the early phase? Any insights into that? And I'll ask that to Nilika, sorry, and I'll widen it.
Yes. So if I can just talk about the Sri Lankan situation. I mean, in the beginning of the dengue epidemic in 1989, our dengue mortality was 9%, but now it has come down to 0.3%. In 2010, it was 1%, and what really made it come down from 1% to less than 0.3% was early healthcare. And that is in the absence of any of these drugs that it was reduced to less than 0.3%. And if you take a lot of other countries who are managing dengue well, the mortality rates are very low. That is because getting people down to hospital early. So whatever strategy is to detect the fluid leakage very early so that you give timely fluid management so that people don't go into severe critical fluid leakage, and then, it goes, it's a vicious cycle, that leads to that, and then subsequently it goes on. So it's actually to prevent all of those happening. And so far, we have been able to, even in Singapore, everywhere, the mortality rates less than 0.3%. The countries where you do see high mortality rates, for instance, there was a recent publication from India that the mortality rates are like 2.6% in some areas, and this is because of delayed presentation to hospital. So I think it is very important that the public health and the dengue control unit or whatever in that country has a strong public health communication strategy to make sure that the patients come into hospital early so that timely interventions can be done. And so in Sri Lanka, we have already sort of got that mindset in our population. So because of that, we do see for the drug trials we do, if you give it early, it makes a difference than giving it later. Thank you.
Great answer. Thank you very, very much for that. I could open that to Kevin and Priya. Any insights into that in terms of the lateness of patients coming in and the effects of any of this on that? Priya, would you?
Yeah, I mean, obviously, if you want to tackle that early phase of viremia, the earlier the better, right? And so encouraging patients to come in sooner rather than later, especially if you know that dengue is peaking in your geographic area, through public health messaging is going to be really, really helpful, right? And then in addition to that, I think, what we've seen with COVID-19 is that now there's an infrastructure in place for community surveillance, right? And so, taking advantage of some of that infrastructure after COVID subsides to redirect that to diseases that are more relevant for those communities will be really valuable and could potentially help indicate when patients need to be more vigilant and alert about these types of things. And so yeah, the earlier the better, especially if you actually want to help the patient showing up to the hospital as opposed to, preventing transmission to caretakers. And so those are the things that I would be thinking about.
Thank you for that, Priya. Kevin, any insight into that, or in terms of your diagnostic, the road to diagnostics? We've been asked questions in terms of,
Yeah, I mean, I think Neelika and Priya really summarized it really well. I mean, what we're studying is probably too early to be useful in community diagnostics. It's maybe more useful when people come to the clinic. I think, Priya really hit it on the head as well. I think getting community diagnostics in place would be really beneficial, particularly in those really rural communities where actually they might not have immediate access to a hospital, but they might have access to a healthcare center, which, will have different levels of resourcing. So, making sure that you can catch people early enough to send them to a place where they can actually get that really crucial clinical management. So I think any diagnostic test would need to really be able to be implemented in settings that are actually not necessarily in a hospital setting, but also in communities that are affected.
Great, thank you for that, Kevin. We've had a question coming from Dr. Aya Maya Win, he wants to know the role of platelet infusion in dengue. Is this an ideal biomarker, or is it really a lab marker for dengue severity? I guess that's the question. So the role of platelet, any insights into the role of platelet infusion within dengue? Is this an ideal biomarker or lab marker for dengue severity? I'll ask that to all three who would like to answer that.
Yeah, so in the early years of dengue, because you have thrombocytopenia and some patients get very severe thrombocytopenia, platelet transfusions were given in Sri Lanka and many other countries. And so later, the usefulness of platelet transfusions were properly evaluated. There was publication, it was published in Lancet also, that platelet transfusions actually don't have any effect at all, and they can actually cause more severe disease. So if I may explain it a little bit more, reduction in platelets or thrombocytopenia appears to be a result of severe dengue and not a cause. So, and that has been shown over and over again. So we have patients who have a platelet, now the normal count is more than 150,000, but patients with even 5,000, 10,000 counts, in the absence of leakage, they seem to be doing fine. Fine in the sense they have body aches and everything, but they don't develop severe disease. They have very low platelets. They don't have bleeding, nor do they have any other issues. So, most dengue-affected countries have actually studied this, and they don't recommend giving platelets. Even our national guidelines and many guidelines, including the WHO guidelines, do not recommend giving platelets because it's actually detrimental and doesn't serve the purpose. And whether platelets can be used as a good biomarker, I mean, platelets do reduction is seen, and if somebody is having dengue, is known to have dengue, or let's say the NS1 is positive. So if the platelet counts are coming crashing down, you would want to actually monitor that patient more carefully, because the platelet lowering of the platelets precedes the vascular leak or accompanies the vascular leak. But having said that, it is not a good biomarker itself of dengue hemorrhagic fever, because there are enough patients who have very low platelet counts, as I said, in the absence of vascular leak. Thank you.
Thank you for that answer, Neelika. I hope that's answered the question I'm sure it has. Any other insights into that from Kevin or Priya before we move to the next kind of question? No? Okay, great. So We heard earlier as well from quite a few of the earlier presentations, and indeed in this session here, similarities between the patients presenting with COVID and some of the clinical presentation of someone with dengue. So I suppose I'm kind of asking the question, you need collaborations with the COVID world in terms of their understanding and your understanding within dengue? Is that starting to happen in your world out there or not? Or I'll ask Kevin that first. There has to be a case made to learn from the. Sorry, yeah.
Well, I don't know the case in other countries, but I mean, I think from the UK research perspective, we've got to remember that SARS-CoV-2 is an entirely new virus. So there wasn't really a COVID-19 world a year ago. So most of the people here who have been studying COVID-19 are actually people who had been studying dengue or Zika or other emerging viruses like Ebola, or there are some people who were already studying coronaviruses. And so a lot of virologists and clinicians have got involved here in the research. And because they're already part of these communities, there has, I think there's lots of opportunities for collaboration. I guess on a slightly different point, I think there are some, great opportunities for learning from both diseases. So actually one of the things that's interesting is, as I said, we've been looking at this parasite interaction to really understand, the molecular events in vascular leakage. But what's interesting is actually that COVID-19, of course, has a big cardiovascular implication. And actually it looks like, in fact, that SARS-2, the virus, actually does have some pretty major effects on parasites in the heart and also in other parts of the body. And we don't know whether that's driving the disease, but it's interesting that, the ACE2 receptor, which is the receptor for SARS-2, is actually probably most highly expressed on parasites actually within different cardiovascular cells. So it's an interesting link to dengue and actually to a lot of other diseases that have affected these interactions or known to affect the interactions, like HIV affects them as well, and Zika and Japanese encephalitis virus. So I think we're very interested in those commonalities between viruses, and actually maybe some of the lessons we learn with dengue might eventually be useful for treating COVID or other diseases that might emerge in the future. Yeah.
That's a very interesting answer, this overexpression of ACE2 on that cell type. Very interesting indeed. Thank you for that, Kevin. Priya, you were nodding. Do you want have you anything to add to that? The COVID world and the dengue kind of world?
Yeah, so I mean, my group is not actively doing a lot of COVID work, but what I will say is that there are some really exciting opportunities now that that people are more focused on viruses and emerging viruses, right? And so one of the things that has come out of this is like the risk of another pandemic, right? And how do we get ahead of that, right? And so what I like to think about from that perspective is, can host targets actually serve as a way to strategically hit multiple viruses families, right? That are genetically unrelated to each other, right? And so by identifying these virus host protein interactions for multiple virus families, diverse virus families, right? We could potentially identify a core group of host proteins to really focus on, right? So that, there are some really exciting researchers that are pursuing how to predict the next pandemic using sort of these types of surveillance methods in zoonosis. But let's say we can't predict that. How can we actually best ensure that we're capable of dealing with whatever comes out, right? If we can predict it, great. But what if we're wrong? Do we have something in our arsenal that could also already work, right? And so I think that's where I'm very excited about all of the new interest in virology and the intersection with dengue and other viruses.
Yeah, that really alluding to the kind of not post-COVID, but the effect of COVID on our lives in terms of this renewed interest, this heightened interest. That's a fascinating kind of dot to resilience, building resilience, preparedness. Well, that's a fantastic answer. Thank you very much for that. We do have a few more questions, well, there are quite a few statements more than questions. So a lot of people are coming back with great presentations, really giving you a kudos in terms of the work that you're involved in. There's a question by Patricia Yulita Gunawan, who's saying, thank you for the great and interesting presentations. The question is, does isoprinosine, is it useful in dengue infection? Some of my colleagues usually use it in dengue. So I'm not sure if anyone wants to answer that or can answer that. It contains methoxyprenol. I'm not sure what or how to, if anybody wants, perhaps we can answer that separately. I'm not sure. There's a question by Belen Pedrick. Great session, thank you. Do we know why post-secondary infections are less severe than secondary infections? What could be the difference in the host response? So I suppose I could ask that to, I'll ask that to Nilika to start with. Do we know why post-secondary infections are less severe than secondary infections? What could be the difference in the host response?
So actually, a secondary dengue infection is defined as an infection after the primary. So with the immunological response, the person has IgM and IgG. So it's, you can't really tell if the person is getting it for the second time, third time, or fourth time based on just that. But given that, but if you think that the IgM and IgG refers to a second time infection, then studies following people up have shown that the person gets it the third time or a tertiary infection is actually less severe than a secondary infection. But having said that, in Sri Lanka, we have seen people who develop a secondary DHF go on to develop DHF again in a very severe manner. So this is why I think, given that DHF, the people who actually develop DHF are a minority, given the whole amount of people who are infected. And if a person gets DHF twice, see, I mean, that's a rarity. So I think there is predisposed host factors that make certain susceptible to DHF, which we're actually looking into. Thank you.
A lot for that answer. Kevin, would you have any input into that in terms of differences in host response? Why secondary infections are less severe than why post-secondary infections are less severe than secondary infections? What could be the difference in the host response?
Yeah, I mean, I think the dogma in the field, which may not necessarily obviously reflect most current thinking in the clinic, has always been that when you have a secondary infection, and it's I guess primarily secondary infection with a different serotype from the first infection. So obviously remember there are four dengue viruses, and there really are four separate different viruses that happen to cross-react antigenically. So when you have that secondary infection, you have antibodies from your first infection, and if you get an infection with a different serotype, those antibodies can bind to the virus, but they don't neutralize it. So they're not protective, but they do actually enhance entry of the virus into immune cells like macrophages and dendritic cells, which actually are where the virus replicates, and that causes a higher viremia and higher replication. And I guess the thinking, at least in the past, was that when you have a third infection or a fourth infection, that at that point you've sort of had a prime boost of infections, right? So your immune response is just much better able to deal with the disease. But as Nilika points out, you know, there is evidence now in various settings, including from Eva Harris's group in Nicaragua, that people can get infected with multiple serotypes, whether or not they present with clinical disease. So, you know, again and again and again. And as Nilika said, some of them are developing severe disease. So I don't know whether those people have a slightly different immune response, but it was always thought that the antibody response after 2 infections was protective, at least from severe disease, if not co-infection. But, you know, whether the thinking on that's changing, obviously, because that was those were quite early studies.
Thank you for that, Kevin, as well. Priya, would you have any insight into that, the differences in host response in terms of why post-secondary infections are less severe than secondary infections?
No, I think Nilika and Kevin really highlighted the salient points there. the only thing I will add is that, the timing between these infections can be a significant factor in determining severity of disease, because antibody immunity can wane, right? And so that's also something to consider. Also, the field, the dogma in the field is very focused on the antibody response, but there's also a T-cell response, right? That we shouldn't forget about, right? And so it could be that some of these events are driven by T-cells as opposed to an antibody response.
Great. Thank you very much for that. Hopefully, Belen, Dr. Pedrick, that's answered your question there. So thank you very much for the panelists for doing that. My final question, I've had a reminder that we're coming to the end of the session. I just wanted to ask actually a non-molecular biology-based question, really. Obviously, we are advocating, and we've been advocating for a World Dengue Day. We're at that point now. I just wanted to ask, really, from your work perspective, your professional perspective, how would a World Dengue Day help you in terms of, would this be useful to you? How would it be useful to you in a nutshell? I think you've got a couple of minutes. So I'll ask that to Kevin first, because he's on our dengue advisory group. So I'd ask you that. In terms of backing a World Dengue Day, if you had to say it in a nutshell, what would you say?
Yes, so I come at it from a research perspective, regardless of whether people are molecular or clinical or translational. And I think for me, one of the things is, people are doing really great work on dengue, but I think what we really need is for people from different parts of the world also to talk, obviously a bit maybe more to each other. And I hope that a World Dengue Day would be a really great forum to connect people from, all across dengue affected regions. And, we don't have dengue in the UK, but I think it will help raise awareness as well here of people who might be interested in contributing. But I think it's really about connecting people and having the reason for everyone to come together. And I think there's some really great regional events and really great to bring various countries together. But I think what we need is everyone to come together across the world. And, Nilika explained really well. And, there's this question of why dengue is more severe in Asia than in the Americas. And I think until you get people from the Americas really collaborating extensively with people from Asia, I don't think we're going to answer that question. And of course, there's this big black box of really what's going on in Africa, where we know there's a lot of dengue and it's not always reflected in the data that we see coming through in the reporting. So I think bringing everyone together to the table from all of the affected regions, I think is really important. And I think you need something like a global initiative like World Dengue Day to make that happen.
Fantastic answer, Kevin. Thank you for that. We've got the Ethiopian Centers for Drug, the CDC from Ethiopia on the next session in terms of their kind of take on it, and completely echo what you're saying in terms of the underreporting in areas like Africa. We need an event horizon, and we believe the World Dengue Day is that. Great answer. Nilika, could I ask you for your input into that in terms of a World Dengue Day, what it would mean to you? We've got in a few words on that.
Yeah, so dengue, the imports of dengue to countries like Sri Lanka is very obvious, and so South Asia, Southeast Asia, and the tropics, which are poorer countries, have been affected by dengue for years and years. And so a World Dengue Day would actually create awareness among other countries about the potential threat, and actually, okay, this is a big problem in these countries. And as Kevin said, it would be really great to collaborate. And actually, we just started to collaborate with Latin America Hepatitis group in doing a comparative epidemiological and immune correlate study between Asia and Latin America to answer that key question of why is it different between Latin America and Asia, or actually is it different when you use the same parameters? That is one important thing we need to find out. But also, like in countries like ours, the funding for dengue is sort of non-existent. So hopefully, with greater awareness, there will be more funding from the countries which do have funding, funds to sort of funding dengue research in poorer countries. So that could be one way that this might help. Thank you.
Great answer. We really appreciate that. Funding is a bit of a sore subject here at the moment with the ODA cuts, but the G7 is on. Who knows? They might reverse it. But that's a great point that you've made there, Nilika. Priya, from your University of California, Davis perspective, would a World Dengue Day help?
Absolutely. There's no question about it. When I think about my own research, it has no relevance unless I can interact with clinicians and people who are developing drugs, right? So I think in terms of actually translating these sorts of molecular level insights into things that can improve people's lives, these types of events where you go all the way from, geographic scale phenomenon all the way down into the molecular level detail is really valuable in terms of getting all these people together, right? And mixing these ideas. So from that perspective, these types of events are extremely valuable so that I can see the forest and not just the trees.
Yeah.
Right. I think the other thing from the US, we don't have locally transmitted dengue here right now, but we're going to, right? Like it's only a matter of time. And so right now, most Americans don't really understand why dengue is such a bad disease and why we should care about it. Fortunately, the NIH does, right? So it still tends to be pretty well funded, but it's going to become even more and more important for my country, right? And then just from a personal perspective, over half of my family still lives in India. And so for me, having these types of events that are focused especially on Southeast Asian and South Asian researchers and their work is really exciting to see.
Brilliant. What a great answer. And we really hope to be, the World Dengue Day will give that event horizon. It will be that galvanizing focal point to bring all the different disciplines together. On that note, I'd like to say a huge, huge thank you to all of you, to Priya, to Kevin, to Nilika for joining us from your various geographies at various times. And to have to sit through my not very good Q&A, but I appreciate that you took your time to do that. Some great answers, and I hope the audience have enjoyed that. Some really, hand clapping and great presentations. Thank you. A lot of people asking for your contacts. So you've kind of entered superstar status, I think, with the Southeast Asian research community, but you probably already have. But it's gone into orbit somewhat. I think they're all asking for your contact details. I'll leave that up to you. I'm being asked to come off at the moment because the next session has to be prepared for. So from me, thank you very much. From the IS entity, thank you very much. And everybody who's joined us, the audience, will have a following session coming in the next half an hour. So from me, thank you. Enjoy the rest of your days and enjoy the rest of the 3 days that we've got planned. Thanks a lot. Okay, bye-bye. Bye-bye.